Tangential flow filtration, TFF. Principles, membrane chemistry, mechanisms, and industrial applications. Tangential flow filtration, TFF, also referred to as cross-flow filtration, is an advanced membrane separation technology extensively employed in biotechnology, pharmaceutical manufacturing, food processing, water purification, and nanomaterial production. In recent decades, TFF has become one of the most important downstream processing methods in modern bioprocess engineering because it enables efficient concentration, purification, clarification, and buffer exchange of complex biological products while preserving product integrity and process scalability. The technology is particularly valuable for handling sensitive biomolecules such as proteins, monoclonal antibodies, vaccines, viral vectors, nucleic acids, extracellular vesicles, and cell-based therapeutics. Unlike traditional filtration systems, where the feed stream moves directly toward the filter surface, TFF operates by directing the feed solution tangentially across the membrane surface. This cross-flow motion continuously sweeps retained particles away from the membrane interface, reducing membrane fouling and maintaining stable filtration performance over prolonged operation. As a result, TFF offers substantially improved efficiency, higher throughput, and greater process control compared with conventional dead-end filtration techniques. Fundamental principles of tangential flow filtration. The operating principle of TFF is based on the simultaneous generation of two fluid streams, the permeate and the retentate. During operation, the feed solution is circulated parallel to the membrane surface under controlled pressure conditions. Molecules or particles smaller than the membrane, pore size or molecular weight cutoff, pass through the membrane as the permeate fraction, while larger species are retained and recirculated within the retentate stream. The primary driving force behind membrane permeation is the transmembrane pressure, TMP, which represents the average pressure difference across the membrane. TMP directly influences filtration flux and separation efficiency. However, the relationship between TMP and filtration performance is highly sensitive. Excessively high TMP can compress the concentration polarization layer or gel layer formed at the membrane surface, reducing permeate flow and accelerating membrane fouling. Conversely, insufficient TMP lowers filtration productivity and increases processing time. Cross-flow velocity, often referred to as cross-flow flux, CFF, is another critical operating parameter. The tangential movement of the fluid creates hydrodynamic shear forces that minimize particle accumulation and reduce concentration polarization near the membrane interface. Nevertheless, excessively high cross-flow velocities may generate shear stress capable of damaging fragile biological products such as proteins, viral particles, lipid nanoparticles, or living cells. Consequently, optimization of TFF requires careful balancing of TMP and cross-flow conditions to maximize productivity while preserving biological activity and product quality. Membrane chemistry and filter materials. Membrane chemistry plays a central role in determining filtration selectivity, fouling resistance, chemical compatibility, permeability, and product recovery. Modern TFF systems employ both polymeric and ceramic membrane materials depending on the process requirements and operating environment. Polymeric membranes are most widely used in biotechnology and pharmaceutical manufacturing due to their flexibility, affordability, and broad range of separation characteristics. Common membrane materials include polyether sulfone, PES, regenerated cellulose, RC, polyvinylidene fluoride, PVDF, polysulfone, and cellulose acetate, Polyether sulfone membranes are particularly popular because of their high permeability, wide pH tolerance, thermal stability, and relatively low protein adsorption. Regenerated cellulose membranes are favored for applications involving delicate biomolecules because they exhibit exceptionally low, non-specific binding and excellent compatibility with proteins and nucleic acids. Polyvinylidene fluoride membranes provide superior chemical resistance and mechanical durability, making them suitable for harsh industrial environments. Ceramic membranes, commonly fabricated from alumina, zirconia, or titania, are employed in applications involving aggressive solvents, extreme pH conditions, or elevated temperatures. Although ceramic systems are generally more expensive than polymeric alternatives, they offer outstanding mechanical strength, long operational lifetimes, and resistance to chemical degradation. These characteristics make ceramic membranes valuable in industrial chemical processing and wastewater treatment operations. TFF membranes are further categorized according to pore size and separation range. Microfiltration membranes typically possess pore sizes between approximately 0.1 and 10 micrometers and are used for cell harvesting, clarification, and microbial removal. Ultrafiltration membranes are characterized by molecular weight cutoffs ranging from roughly 1 to 1,000 kilodaltons and are commonly employed for protein concentration, vaccine purification, and viral processing. Nanofiltration and reverse osmosis membranes enable the removal of small organic molecules, salts, and dissolved contaminants in water treatment and specialty chemical manufacturing. Filtration mechanism and fouling behavior. One of the defining advantages of TFF is its ability to reduce membrane fouling relative to conventional filtration systems. In traditional dead-end filtration, suspended particles accumulate directly on the membrane surface, rapidly forming a dense filter cake that increases hydraulic resistance and decreases filtration efficiency. Over time, this accumulation severely restricts fluid flow and necessitates frequent cleaning or membrane replacement. In contrast, TFF minimizes particle buildup by continuously sweeping retained materials along the membrane surface through tangential flow. This dynamic hydrodynamic environment reduces the rate of cake formation and maintains relatively stable permeate flux during operation. Nevertheless, membrane fouling remains an important challenge in TFF processes, particularly in high-concentration biological systems. Fouling mechanisms in TFF include pore blocking, adsorption of proteins or macromolecules onto the membrane surface, gel layer formation, and concentration polarization. Concentration polarization occurs when retained solutes accumulate near the membrane interface faster than they can diffuse back into the bulk solution. This localized enrichment creates additional osmotic resistance that lowers permeate flux and may ultimately contribute to irreversible fouling. Modern membrane engineering strategies have significantly improved fouling resistance through surface modification techniques, hydrophilic coatings, zwitterionic surface chemistries, and low-binding membrane materials. In addition, optimized flow channel geometries and automated process control systems help maintain favorable hydrodynamic conditions that reduce fouling and extend membrane lifespan. Differences between tangential flow filtration and traditional filtration. Tangential flow filtration differs fundamentally from conventional dead-end filtration in both flow orientation and operational behavior. In dead-end filtration, the feed stream is directed perpendicular to the membrane surface, causing retained particles to accumulate directly on the filter medium. As the filter cake thickens, resistance to flow increases continuously, reducing throughput and requiring periodic interruption for cleaning or filter replacement. By contrast, TFF directs the feed stream parallel to the membrane surface, allowing retained particles to remain suspended within the recirculating retentate stream. This cross-flow configuration substantially reduces membrane clogging, improves operational stability, and enables continuous processing over extended durations. Consequently, TFF systems are better suited for high-solid suspensions, concentrated biological streams, and large-scale industrial manufacturing. Another major distinction lies in process scalability and automation. Traditional filtration methods are often limited in their ability to maintain consistent performance during scale-up, particularly in biopharmaceutical applications involving sensitive products. TFF systems, however, can be scaled effectively from laboratory benchtop systems to pilot-scale and industrial manufacturing operations while maintaining similar hydrodynamic conditions and separation characteristics. Automation and modern TFF systems. Early TFF operations often relied on manual or semi-automated systems assembled from independent pumps, valves, tubing, gauges, sensors, and reservoirs sourced from multiple suppliers. Such systems required constant operator supervision to maintain pressure settings, monitor flow rates, document process conditions, and ensure consistent performance. Modern automated TFF systems have transformed bioprocessing by integrating programmable control systems, digital pressure monitoring, automated valves, flow sensors, and real-time data acquisition software into unified platforms. These advanced systems enable highly reproducible operation, minimize operator intervention, and improve compliance with regulatory standards such as Current Good Manufacturing Practice, CGMP. Precise control of TMP and cross-flow velocity is especially important during the manufacture of biologics and advanced therapeutics. Improper combinations of pump flow rates, tubing dimensions, or membrane configurations can generate cavitation, excessive fluid velocities, or pressure fluctuations that damage sensitive biomolecules and cellular products. Automated TFF systems provide accurate monitoring and control of these parameters, reducing operational errors and enhancing product consistency during process development and scale-up. Industrial and biotechnological applications. Tangential flow filtration has become indispensable across numerous modern industries. In biopharmaceutical manufacturing, TFF is widely used for concentration and diafiltration of monoclonal antibodies, recombinant proteins, vaccines, and gene therapy vectors. Ultrafiltration and diafiltration processes enable the removal of impurities, the exchange of formulation buffers, and the concentration of therapeutic products prior to final formulation and filling operations. Vaccine manufacturing extensively relies on TFF for purification and concentration of viral particles while preserving biological activity. Similarly, gene therapy production uses TFF for processing adeno-associated virus, AAV, vectors, and other viral delivery systems. In cell therapy and regenerative medicine, TFF facilitates gentle processing of cells and extracellular vesicles under sterile and closed system conditions. Outside biotechnology, TFF is widely used in food and beverage processing for whey protein concentration, milk fractionation, juice clarification, and microbial stabilization. Water treatment industries employ cross-flow membrane systems for desalination, wastewater recycling, and contaminant removal. Nanotechnology applications include purification of nanoparticles, liposomes, colloidal dispersions, and advanced nanomaterials. Emerging applications of TFF continue to expand in areas such as continuous bioprocessing, mRNA vaccine manufacturing, exosome purification, and advanced biomanufacturing platforms, highlighting the growing importance of membrane-based separation technologies in modern industry. Advantages and technological significance of TFF. The widespread adoption of tangential flow filtration arises from its ability to combine high separation efficiency with gentle product handling and scalable operation. By reducing membrane fouling and maintaining stable permeate flux, TFF enables longer operational cycles and improved process productivity. The technology also offers superior product recovery compared with conventional filtration methods, minimizing loss of valuable biomolecules during processing. The ability to automate TFF operations further enhances reproducibility, process consistency, and data integrity, while reducing labor requirements and operator-dependent variability. These advantages are particularly critical in regulated pharmaceutical manufacturing environments where product quality and process traceability are essential. Because of its versatility, scalability, and compatibility with sensitive biological products, TFF has become a foundational technology in modern biotechnology, pharmaceutical engineering, water treatment, and advanced materials processing.